Now, a little bit of background on Anna. She originally grew up in Australia, came to Colorado as a high school student. After a couple of extended time in college, she ended up at UNC. She was actually in the very first course that I taught her. So imagine less gray hair, some of which she has imparted upon me. And Anna initially did a master's degree working in my lab, doing a conservation genetic study on cacti, and had a defense similar to this in 2013, I think, 2012, a good long while ago. Went off and started working in plant conservation field, did research at the Denver Botanic Garden. But whenever I would run into Anna, she kept on mentioning, we've got this new thing going on in the state, cannabis. There's all sorts of things that are screwed up. We understand plant genetics. We can research this. And every time I would say, Anna, I do plant conservation genetics, not pot. I don't think I want to go down this road. And for about a year, she kept on poking, talking about examples, and making a good case for the fact that there are lots of interesting questions out there. And since we're dealing with an organism that was previously illegal and has now come into this pseudo-legal world, which she'll talk a little more about, that there's all sorts of basic biology that we just don't understand. And for the vast majority of organisms, that's not true. And that's because we can't grow cannabis here. Well, we can grow hemp, but there's lots of limitations upon what we can do. So working with Anna on this project has been an education for me, related to new things. As her talk will show, it's been a really good example of collaboration and reaching out to other people in order to answer various questions. Anna's research has been written up in Wired magazine, has been written up all over the cannabis literature. We've attended lots of conferences. She's now definitely an expert in the field of thinking about cannabis genetics. And it really shows an example of how you can go out and stake your own way if you're dedicated and you want to work hard. So it is my pleasure to introduce Anna Schwabe. It's been great. She actually has been the longest running fixture in my lab in the whole time that I've been here. So this time she has to leave permanently, but I'm going to be really sad to make her go with her. So Anna, take it away.
Hi everybody, I am so excited to be here, and so I'm just going to kind of dive right in and tell you how and why I am interested in cannabis. So, here's me, sitting around with some friends drinking coffee, and one of my friends mentioned that now that, back in the 2012, now that cannabis is legal, she's found this wonderful strain. It does everything that she needs. She can get through her day without anxiety or whatever. But wherever she went and tried to get this strain at other dispensaries, it wasn't the same. And I thought, well, that's weird. Like, if you go to the store and you buy a Granny Smith apple, there's an expectation of what your Granny Smith apple is going to be like, right? So you're going to go, you expect a green apple to be crisp and tart, right? And I'm thinking, well, what if there's this situation going on with cannabis? Do we have any way to check that? And we have all of these different kinds of apples. We have all of these different kinds of cannabis. So I thought this would be really interesting to look at and from a genetic standpoint. And so, this is the overview of kind of what I'm going to talk about with you today. So I'll give you a little bit of background in terminology. There's lots of words that are used in cannabis that aren't necessarily transferable across to other plants. So I'll give you a little bit about that. I'll tell you about sources of variation. I'll go through genetic variation, so this is where my study started. And I started with variation within strains. And then I expanded it to look at the different groups. And these different groups included cannabis that was provided by the National Institute on Drug Abuse, which will be abbreviated as NIDA throughout the talk. And then I looked at some sensory variation, cannabinoid variation, and terpene variation. All right, so background and terminology. So here is the current status of cannabis legalization throughout the globe. So the green areas are where medical is allowed, and you can see up in the United States there's a bunch of states that have green. And then there's now a couple of countries that are starting to do recreational as well, and we've all heard Canada's legalized cannabis, and there's other countries that are kind of following suit. And here's the current status in the United States. So everywhere where we've got recreational as legal cannabis, we also have these other two categories that go along with that. Medical is CBD, so anywhere where it's medical, they allow CBD. And then some states only allow CBD, which is an extract from the plant that can be used as a medicine. All right, so let's talk about cannabis. So currently, the taxonomy of cannabis, Sativa, in the genus Cannabis, it's the only species that is recognized. So you've heard about hemp and you've heard about marijuana. It's all Cannabis Sativa, so one species. It's highly versatile, and I like to refer to the 5 Fs, and that would be food, fuel, fiber, fun, and pharmaceuticals. I'm not a great speller, but I think that's fine. So common differences. So we've all heard about hemp, and the country just legalized hemp across all 50 states. Hemp is defined only by the content of THC, and it's less than 0.3% THC would be classified as hemp. And generally, the hemp types we're going to use for food, so the seeds, maybe fuel, fiber, and that's generally what we think about when we think about hemp. It can also apply a little bit to pharmaceuticals, but we've got the other drug types, which are commonly referred to as marijuana. Those have more than 0.3% THC, and generally, if you go to a dispensary, you're going to have something that's upwards of 10% THC. And then we have these groups here, which are very commonly talked about among consumers and customers and just general public. Sativa, indica, we have a hybrid of those two. And then there's an up-and-comer called that's this high CBD type, which has a higher level of CBD, but also has maybe a bit more THC than what we would allow it to be called hemp. So people are kind of paying a little bit more attention to this. All right, so these are so the plant produces what are called cannabinoids, and these are two of the cannabinoids that are commonly talked about. The plant actually produces an acidic form of this, and through certain processes, you can decarboxylate the acidic forms into the bioactive forms, which we refer to as THC and CBD. So THC is generally the fun and pharmaceutical cannabinoid. That's the one that gets you high, right? You can also use it for things like relaxation. If you're having trouble sleeping, you might be able to use it to alleviate that. You can use it as an appetite stimulant, so we've all heard of the munchies. And then inflammation and pain relief. And then CBD is generally thought of as a pharmaceutical cannabinoid, and so it's known to relieve pain and inflammation. It will not get you high, so if you take CBD oil, for example, you're not going to get all weak. And then it's known to suppress seizures, combat anxiety, reduce nausea, and it can be used as an antidepressant. So that's just two of the cannabinoids that are fairly commonly referred to in the media and whatnot. All right, so what are we looking at here? So variation sources. So phenotype equals environment plus genotype. So phenotype. Phenotype are the characters that we can observe on the outside of the plant. So things like the cannabinoids, so the chemicals of the plant, they have different effects on us. We can smell the terpenes, and the terpenes are what give the different flavors and aromas. It also refers to the morphological characters, so things like leaf shape, leaf size, is it fat, leaflets, is it dense branching, dense flowers, things like that. We have the environment, so how was the plant grown, when was it harvested, how was it cured, how long was it in storage. All of those are outside conditions that can have an effect on the phenotype of the plant. So if you change any of these things, you give it more nutrients, you grow it in different kinds of soil, you give it more water, less water, you're going to end up with something that's a little bit different than something that's grown under different conditions. So that feeds into the phenotype. And then we have genotype. So this is the blueprint for the organism, and generally this doesn't change. So DNA, chromosomes, alleles, loci, the whole genome, all of that is part of the genotype, and that also, of course, feeds into the phenotype. So, if we can verify the genotype, and if we can standardize the growing conditions, harvesting conditions, storage conditions, potentially we can decrease variation in phenotype. And so really that's what I'm looking for. In all of these things that I've done, I'm looking for what's causing that variation that I first observed when I first heard about the difference among dispensaries. So here's some of the sources of genetic variation. We can have slight variation when we have seeds from plants because they've been pollinated usually, and not super going to go into that because it's pretty complex. Underground trading, so it's been illegal for a long time, but people have been trading plants, like, hey, I'll give you a little snippet of mine if you give me a little snippet of yours. And then that can possibly lead to mislabeling. So I forgot what Steve said this is. He said it's purple. I'll just call it purple cloud. That's it. But it's not, right? Steve had a name for his stuff, and you just renamed it. But it would have the same genotype. And then, of course, mistaken identity. Well, it looks and it smells and it feels like Blue Dream, so we'll just call it Blue Dream, right? And then renaming and relabeling, and that could possibly be at a dispensary. They're trying to move product. They've got something that isn't flying off the shelf, so put a Blue Dream label on it. Bam, it's gone. All right, so this is kind of a problem, I think, for the industry. We've got this brand new industry. Everybody's trying to work everything out. But currently we have no strain verification available, so there's no database that will tell you what strain you have based on anything, really. We rely on experts who can tell what things are. There's no protection for developers, so these breeders that are coming up with new strains with characters that are desirable, there's no intellectual property protection for these guys. They can't register their strains or their varietals or their cultivar or any of that. And so that goes for new or vintage strains. So on the consumer side of things, consumers are being provided inconsistent products, potentially having undesirable effects. You can imagine if you took an aspirin for a headache, but somebody gave you a Benadryl and then you fall asleep at your desk, you're going to be not real happy. Plus you still have it, right? It's not verifiable. They just have to go with, well, he said it was Blue Dream. I bought Blue Dream. I got home. This doesn't feel like Blue Dream. But they can't verify it. And of course, there's no protection. So you can't go back to the store with your little Blue Dream. This is Blue Dream. So there's a little bit of a problem here. So I came up with some research questions. So this is kind of how my research went, and I'll tell you the evolution of my research, because this is not at all what it started out as. I wanted to examine the genetic integrity within strains. I wanted to examine the genetic relationships among categories, so those would be feral or escapees from cultivation and then cultivated hemp. And then the drug types, so high CBD, indica, sativa, hybrids. And then I also wanted to determine how differences in genotype are reflected in the chemotype. And so things that go into the chemotype are going to be reflected in aromas, because those terpenes are what gives the plant the smell. The cannabinoids, which are those things that have effects on the body. And then terpenes, again, those are the things that give the smell, but we can measure the terpenes and actually see which ones are in there. So, again, I'm just going to remind you that this is the equation that I'm looking at, and with my expertise, I can do the genetic portion of this. So, genetic variation part 1, within strain variation. So my sampling. So I went at this from a customer point of view. So I went into dispensaries and I said, I'll have a gram of that and a gram of that and a gram of that. Paid for it. Didn't tell them who I was. Didn't tell them that their weed was going to be scientifically examined. So they had no idea what I was doing with it. And it was all based on what was available at the time when I went in. And I got the strain information from online databases such as Leafly and Wikipedia. Not Wikipedia, Wikipedia. Okay, so a little bit about this indica and sativa thing that people always talk about. So here's my sampling, and these are arranged from 100% sativa, so based on stuff that I got online. going through down to 0% sativa. So these are 100% indica. Does that make sense? Cool. So these ones in the middle are going to be a 50-50 split of what is considered sativa and what is considered an indica. So sativas are thought to be these uplifting strains. They have narrower leaflets, they're taller, more spindly, uplifting effects, sort of party, or anxiety-inducing, so highly drugs and then indicas on the other end of the spectrum, thought to be sort of, relaxing, couch lock, video games, and they're denser branches, fatter leaflets, darker in color. And then the hybrids, of course, are a cross sort of in between those two. So I got 122 samples. I went to 3 different states, 20 different dispensaries, 8 different cities, and in Colorado, I sort of went all around northern Colorado because that's where I am. And so this is what I did. So I did some genetic analyses on these samples that I got, and I used 10 microsatellites. And if you don't know what a microsatellite is, that's fine. All you need to know is that it's a repeating unit, so those letters in the DNA, and they're repeated and they're highly mutable. So here you can see 3 samples of purple kush from different dispensaries. This is kind of what I'm looking for. So here you can see these 2 purple kushes have the same size microsatellite fragments, so the same number of repeating units. But this one right here is missing one. So it's a 3, it's like AGT, it's missing one of those units. So it's 3 base pairs shorter than these 2. So that's kind of what I'm looking for. And then we can compare across all these samples and across all of these microsatellites, and we can determine who's most related to who. And really what I'm looking for, as far as within strain variations, I'm looking for these things to be identical. Anytime they deviate from that, that tells me that there's variability within strains. So here's a clustering analysis. And so I've arranged this again from 100% sativa down to 0. So these are indicas. Greens are in the middle. If there was anything to this hybrid indica sativa grouping, you might see some of the hot colors grouping together and the cool colors grouping in another spot. Or it could be a gradient ranging from hot colors all the way through cool colors. And we're not seeing that. We're just seeing everything kind of flip. We do see some clustering for strains, so we have German poison up there and blue dream down here. But we also see things that are outside of their cluster, indicating that there is variation within these strains. So then I wanted to look at the structure in these strains, the genetic structure. And so I did, this is a Bayesian cluster. This is another kind of clustering analysis where it's going to assign genetic variation to clusters. And in order to figure out how many clusters I had, I ran a likelihood clustering using the Avano method to tell me which one was the most likely, basically. And it came up with K equals 2. So the number of genetic groups for my data was 2. And I thought, awesome, indica sativa, got it. Well, that's not what happened. So these individuals, so each bar is one individual. They get assigned to the 2 genotypes that I most likely have. And the genotypes, it doesn't really matter what that means. But one individual has an assignment to one or the other, or a little bit of both. So for example, if we had an individual like this, they have 30% assignment to genotype 1, 70% assignment to genotype 2. And what we would expect is within a strain, you would expect all of the samples within the strain to be very similar, which is great, sometimes. So ChemDog has a pretty nice, stable signal that's mostly yellow and a little bit blue. We've got German poison up here, which is pretty good. All of them look almost exactly the same, except for this one right here, which decided that it's something else. Or someone else decided that it was. And then we have Girl Scout cookie, which is just all over the place. And I can't figure out what its most likely genotype is. I need more sampling for that. So yes, we have within strain variation. So that's interesting. So basically my conclusions for this portion of my study was that The sativa hybrid indica are not genetically well defined for this data set. There is variation within strains, which could be evidence of mislabeling, misidentification, or relabeling. And there are some strains that are cohesive. So for Jack Flash and Island Sweet Skunk, all of the samples, which are 2 and 3, were genetically very similar. And Ken Dog is pretty good as well. And then Blue Dream and Durban Poison, 9 samples, only one of those samples fell outside what the norm for the other ones were. But 27 strains had at least one genetic outlier, which says that, yeah, there's variation within strains. Enough with some of these smaller sample sizes to say, this should be looked at. So then I went to the Institute of Cannabis Research conference and I presented this data that I had that I was so excited about. And I met Daniela Vergara, and she tweeted out that she was doing cannabis genetics using the whole genome, and we were kind of finding the same thing, so that was interesting. And then it's also where I became aware of this NIDA stuff. Daniela did a chemotype analysis and found that the stuff that the government was growing, that the National Institute of Drug Abuse, the stuff that they're growing at the University of Mississippi, and I'll go into that in a sec, is quite different chemically from what you can buy in any of the retail markets. And Sue Sicily ordered some NIDA weed for her PTSD studies, opened the packet and was really shocked at what she dumped out, which was this stuff. And she said it didn't smell like marijuana, like not what you would expect, because it's usually pretty smelling. And some of it was moldy. She was just like, what is this? This is research grade marijuana. And so I was like, well, I can figure out what this is. So a little bit of background on the National Institute of Drug Abuse. So they provide research grade marijuana for cannabis studies in the United States. It's controlled by the DEA and it's grown at the University of Mississippi. And as far as I can tell, the last time they harvested was 2014. I haven't been able to find anything new. So they're kind of giving old weed for research. And Daniela's results do suggest that. And so they have a mission statement here. They want to advance science on the causes and consequences of drug use and addiction and apply that knowledge to improve individual and public health. So they've got an agenda a little bit. So I decided to check it out. So I'm looking at the whole cannabis spectrum, so all the usage groups, and I wanted to see where on that spectrum NIDA was falling. So I collected some feral hemp, so wild collected hemp, and I got those samples from various herbaria around here in the Denver Botanic Gardens, and I had some farmers send me some cultivated hemp, and then of course I had access to the drug types, and I got a couple of high CBD types that were provided by some nice farmers, and I also had access to NIDA research grade marijuana because our chemistry department has access to that, and so that's where I got the DNA for this. So here's my sampling, and you can see that I only have 2 NIDA samples because really they don't have a whole lot on offer. 3 high CBD strains, and then a bunch of drug type strains, and then some hemp for comparison. And so sort of here's where I got the stuff from. And Richard Hyslop in the chemistry department was nice enough to let us use some of the DNA from some of his samples from NIDA. So here's what we found. So first I looked at divergence. So this is just genetic divergence between 2 groups. So I know this table is kind of like, but if you look, all we're doing here is we're comparing one group to another group. So to orient you here, this is called FST value. It's a divergence statistic. The lower the number, the more closely related the 2 groups are. The higher the number, the more diverged the groups are. And so as we go down this list here, we see hybrid to indica and sativa. We would totally expect that since hybrids are a cross between the indica and sativa. You move down the list, and the numbers start getting higher. Well, what's a bad high number? What's a good low number? So for context, FST value of 0.15 can be used as a species delineation as species are, within species are diverging. So 0.15 is sort of the thing where you go, Yes, they are different enough we can name them something else. So we get down to NIDA versus hemp, and we start seeing numbers like 0.234, 0.363. Those numbers are quite high for within species divergence. So that was pretty interesting. So then I did structure, so this is the same exact thing that I showed you before. We've got 2 groups up here, which was our most likely number of groups in this analysis. But I also did 3, just to show you that there is more signal in the drug types. So it's arranged hemp up here. The next group is NIDA. Then we have high CBD, and then we have Sativa, hybrid, and indica. And then same thing with the K equals 3, same thing. But what we can notice here is that there is a very strong signal that kind of belongs to hemp. There is some drug types that do have a strong hemp signal, but that's okay. They're all the same species. We kind of expect that. But what we can also notice is that this very strong signal that's associated with hemp is really strong in these NIDA samples. And if we pull it out to K equals 3, we can see there's more of this purple signal now in the drug types indicating a little bit more diversity. But we still have this really strong signal in these NIDA samples that still are really strongly associated with those hemp samples, which is super interesting. So then I did a clustering analysis just to back up my thoughts. And you can see here that we've got these drug types. They are all overlapping, clustering together. We've got a nice hemp group here with NIDA nestled right in the middle of that cluster. And if you're having a hard time visualizing all the different colors, drug type is green, hemp is yellow, there's NIDA right there. All right, so the conclusions for this portion of the study are that hemp and drug types are genetically distinct. Sativa and indica hybrid, still not super clear from any of these results. Hemp strains have less genetic variation than the drug types. And the most important thing I think about this study is that NIDA research-grade marijuana is genetically distinct from those drug types that people can actually get their hands on. And it's more similar to hemp. So, I mean, we've got some broader impact here, right? So all this research on marijuana and smoking marijuana, and does it do anything for your health, and does it help with this and that? Probably not if you're going to use that. So this review that I came across, one of their conclusions was that most trials showed improvement in symptoms associated with cannabinoids. These associations did not reach statistical significance in all trials. Well, yeah. I probably wouldn't agree. I would disagree with that because, take away from that what you will. So May of last year, I decided to put up that first study, wrote it up, and put it up as a preprint on bioRxiv. bioRxiv is not yet peer-reviewed. However, this study has been peer-reviewed since and has been accepted into the Journal of Cannabis Research. They just haven't published it yet. And then Felipe Henry in Canada, he was excited about this, and he actually emailed me. We talked for a little bit. But this guy also got a hold of it. His name is Avery Gilbert, and he is a scent scientist, for lack of a better title. But he published this study, Consumer Perceptions of Strain Differences in Cannabis Aroma, last year. And when he got in touch with me, I was like, totally know where you want to go with this. This is exciting. So we decided to do a collaboration on genotype versus phenotype. So going back to that equation again, we've got aroma, cannabinoids, terpenes, all feeding into that phenotype. And then we have the genotype. What if we knew the genotypes and then could look at that? We could potentially eliminate this portion of the equation and have phenotype equals environment. So pretty cool. So we decided to do this cannabis smell study. You might have seen flyers up around campus or whatever. But we chose widely available strains, popular strains that we knew from Avery's results that had different smells that people could detect. Sammy and I went around and all over northern Colorado, even out to Breckenridge, and we picked up a whole bunch of samples, 20 21 dispensary. That's not even all the dispensaries, that's just the ones we chose. And we did genetic analysis. And what we found was, for OG Kush in our sampling, we had all of these samples were genetically identical, and we had one outlier, so perfect. Same thing with Blue Dream, we had one really different one. Mob Boss, we had one that was different. And then Durban Poison, we didn't have enough of this sample to use it in the sensory study, but what we did have was 3 identical samples that we could potentially use as a control, because they're all identical. So we threw those in to the study. And then Sour Diesel, we didn't find an outlier, so that got tossed out as a strain that we could use for the sensory analysis. So clustering analysis backed up what we thought. We've got outliers, so you can see here Blue Dream's clustering, and right here is the outlier. OG Kush is clustering, out here is the outlier. So that just told us, yeah, we've got good sampling, good outliers, and we can go ahead with our study. So we put them in bottles and baggies with numbers on them. So there's no identifying information on these bags of weed. So Avery didn't know what they were, and we were able to recruit 55 participants for this study. They were issued an iPad, and we had 40 odor descriptors. So participants would smell, and then they would do a check-all that applied for everything that they could smell in that sample. And we had the control, which was those 3 Durban Poisons that were the same. We had OG Kush with 1 outlier, Blue Dream with 1 outlier, and then Mob Boss with an outlier. And so here's the 40 odor descriptors that the participants had to choose from. And you don't have to read all of them, but you get the idea. So this is some of the data analysis. So this is the 4 different strains. These do not include the outlier. So these are just the consensus samples, and I'll refer to them as the genetic consensus samples throughout the talk. So those are the ones that were genetically really similar or identical. So you can see, just by looking at this, that we have variation among these samples that were genetically cohesive. We also have some that are very similar across those cohesive samples, but there's definitely variation. So this, we needed some more information. We needed to look at this a little better. And so what we did was we pooled the top 5 odor descriptors for each sample, and then looked at the mean of the genetic consensus samples and compared that to the level of the outlier for those smells. So you can see that there are some where the consensus samples smelled very similar to those consensus samples, but also you can see that, for example, Blue Dream was much more pungent, chemical, and skunk. Online information says that Blue Dream is supposed to be berry and sweet and nice, and those pungent notes don't really jive with what Blue Dream is supposed to be, which tells us that, yes, the outlier is quite different from what it's supposed to be. And we had the same thing with Mob Boss, so that it had a lot less of these earthy, woody chemical smells than the consensus samples. And then OG Kush had this weird cheese smell in the outlier. And according to Avery, that's not really something that people generally pick up on very often because, ew. That's all I can say about that. So I wanted to bring the data down to a more manageable size. So what I did was I grouped the scents, and then I was able to calculate the frequency based on how many times it was smelled, and come up with a little better an analysis, I guess. So I grouped these things. This is my grouping, and I call these perceived shared characters. And so I have an earthy category, and so these are the sorts of things that I put in that category, so coffee and roasted smells and nutty smells. Spicy was like dried leaves, and then spices, of course. Sweet was honey and fruits. Floral, of course, is flowers, and also fresh leaves. And then the pungent category is all the yucky things you don't really associate with smelling nice. So ammonia and gas and skunks and things like that. Cheese. So if we look at the analysis from the shared character, perceived shared character states, this is what we came up with. So you can see here, Blue Dream, really similar in these 4 categories, super different in the pungent category. Mobboss definitely had some differences between the genetic consensus sample mean and then the outlier. And OG Kush is all over the place, but less sweet and more floral and earthy. So I was like, well, that's great, but I'm not a scent scientist, so maybe my groupings aren't accurate. Like maybe somebody else has come up with something better. Well, there really isn't much out there, but I did come across 2 flavor wheels online that describe cannabis smells, and this is kind of the groups that they put them into. And so we've got nice smells, sweet smells, which would be fruits and flowers, and then we've got sour smells, so citrusy stuff, cheese is in that category, spices, and then bitter, which I don't know any other way to describe this other than manly scents. And when I analyzed this, I sort of saw the same thing. So we've got something that's a lot more bitter in the outlier than in the consensus samples. Mobboss was definitely more sweet and a little less bitter and spicy. And then OG Kush was pretty great across the board except for this bitter category. The outlier was less bitter. And so to conclude, here's what we saw. So this is the outlier, and sort of this is an agreement. Flowery, sweet, sweet, that's an agreement. And OG Kush is just a weirdo. It doesn't agree with anything, and it smells like cheese. At least the outlier does. So there's variation in the sense that people detect within strains. The genetic outliers are perceived as different, which is interesting. And yeah, ew, cheese. Oh, sorry. So then I wanted to look at the phytochemical variation, cannabinoids. So what I did is I took those samples that we had used in the sensory analysis, and I got in my car, and I drove them over to Mile High Labs, and they did the analysis for me. So what we looked at was cannabinoid levels. So cannabinoids are over here. As I said, the acidic form is what's produced in the plant. So you see that in an abundance in the plant, and then you see these other cannabinoids listed here, and much smaller amounts of those, and I'll call those the minor cannabinoids. But you can see, here's the outlier, and it looks good with these 2. We know it's different because it smells different, and so I thought, well, if we know that they have different harvest times and curing processes and things, maybe it's a relative abundance thing. Like maybe the ratios of those cannabinoids are the same if we take the total amount of cannabinoids and look at it that way. So I did that. And in some instances, like the THCA here in the German poison, it did level it out. They all had a very similar amount of THCA. But we've got, a pretty big difference between the CBGA. Blue Dream's pretty good. This is supposed to be the outlier. It's red and lime. Here's the outlier for Mobboss, and here's the outlier for OG Kush. So this doesn't really tell us anything other than that there is variation among cannabinoids and within these strains. So then I just looked at THC to see what's the difference there, and again, we don't really see the outlier necessarily standing out as something that contains a different amount of THC than those consensus samples, but the consensus samples also have variation. And then I looked at the minor cannabinoids, same thing, we've got the outlier down here. Mobboss looks very different than this one, which has this THCV, which neither of the other 2. So I was just like, okay, this is a little crazy. Maybe I should look at it the same way I did with the scent and take the consensus sample mean of those ones that we know are genetically similar, compare that to the outlier. So I did that. For Blue Dream, the outlier for THCA was within the variation of the consensus sample, so it was fine. Same thing with Mobboss. And OG Kush, that outlier was outside 1 standard deviation of the mean of the consensus sample, so it was different. And then I did the same thing with the minor cannabinoids, and you can see that for Blue Dream, CBC was outside that standard deviation, THC and CBG for Mobboss were outside the mean of the consensus samples, and CBDA in OG Kush. So the conclusions for this portion tell us that there is variation among genetic consensus samples as far as the cannabinoids go. Some cannabinoids, the outlier varies from the average of the consensus, but there is too much variation in the cannabinoid levels to start making assignments of what might or might not be a strain. So then I decided to look at the terpenes, and we kind of already touched on this with the sensory, but I really wanted to see the actual chemicals and the levels, and how does that relate to the genotype. So I looked at 21 different terpenes, and here they are. These are just the consensus samples. There's no outlier in this, because we already know they're different, so we threw just the consensus samples in here. And then I threw in 3 more strain samples just to show variation among samples. And you can see there's lots of variation among these strains, and it's across the board. So, for example, German poison is much higher in terpenoline than the other samples that we looked at. Here we have German poison because all of those were genetically identical, and you can see that we've got some terpenes that are pretty consistent across the board, but we also have a lot of variation in some of those terpenes. Same thing with Blue Dream. You can see there's not too much variation, but there is variability within the terpenes. And I could squeeze in all 4 graphs on this, but we decided to make it easier for your eyes, that we would just tell you that the same thing happens in Malbos and Kush as well. So here we're looking at the mean of the consensus samples, and then the deviation from that as far as the outliers go. 17 and 21 of the terpenes in Blue Dream were outside of the consensus sample standard deviation mean. 19 and 21 of the terpenes from Malbos were outside of what we would expect. And then even in OG Kush, where there's this huge amount of variation in the terpenes, we had 11 and 21 that were still outside the standard deviation in the consensus. So from that, I conclude that there is variation among genetic consensus samples in relation to terpenes. More than 50% of terpenes in the outlier vary from the average consensus mean, and variation in terpene levels would make strain assignment by terpene difficult and probably unlikely. Phenotype conclusion. So overall, I looked at phenotype 3 different ways, and we know that the phenotype is variable, even when the genotype is consistent. So that tells us, or it should tell us, hopefully it's telling you, that we've got some serious differences in growing conditions, environmental conditions, harvesting times, storage, curing, because there are no standards in the industry yet. So soil, light, nutrients, all that stuff. We can harvest early, mid, late. That's going to change all of those things. And storage conditions, like if you just put it on a shelf in a box, probably not how you should store it. Oh, and we found something else, and this is really exciting. And I just threw this in like a couple of weeks ago, and I was like, oh man, I should look at this. I wanted to look at what they print on the package as far as THC, because they have to do that. They have to tell you how much THC is in the product that you're buying, versus what we measured at Mile High Labs. Most of you. Okay, so what the dispensaries are reporting, what their labs are measuring is in purple. What I measured is the blue. P-value, for those of you who know what a p-value is, it's a significant difference from what they're reporting to what I was able to measure. And I just took the mean of all of these samples, 13.12, and the reported mean, that's huge. And this brings up, or it should bring up, all kinds of questions. Right? We hear about these really high THC strains, 31%. What? Is it? I don't know. So somebody needs to do some more work. By the way, I'm hired. Okay, so conclusions overall. There is genetic variation, could be reflective of mislabeling or misidentification. Hemp and drug types are genetically differentiated. NIDA, research grade marijuana, debunk. People can detect differences in strains, which was interesting and kind of a nice result. But there is aromatic variation within strains. So I don't think you can sit a strain and be like, yeah, that's totally chemdog. There's chemical variation, and that is reflective of genotype, but it's also substantial variation within strains. So I think we need genetic testing. I think we need to have people know what they're starting with as they're growing. Because I think that we also need some environmental standards. We need them to be using, or at least tell them what they're going to get. If they have this kind of soil, this kind of temperature, this kind of water, these kind of nutrients, standard conditions, they can deviate from that if they would like to. But we need something. Because I think that consumers deserve consistency. And so we definitely need more research. And by the way, that's not the way that works. But I think stock photos are super funny, so I figured it out. So with that, I would like to thank my committee members, Mitt McLaughlin, I forgot to turn in Mitt McLaughlin, Nolan Kane, Dr. Heislop, Dr. Reinhold, Connor, my undergraduate, he's not here because he's at a festival in Mexico. Sammy, he's helped me a lot. Avery, Mile High Labs, my friends, family, graduate students, and then the School of Biological Sciences, Natural and Health Sciences, and the UNC Grad School. And with that,
we have about 5 minutes for questions from the audience.